Hello, everyone. Thank you very much for joining us in the third talk from the 2021 Physio webinar. This is an event organized by graduate students from the Physiology graduate program of the University of São Paulo. This is our team. Okay. We organized 4 very interesting talks with speakers from different countries talking about different physiology teams and their intersection with other areas. So please follow us to get more information so you do not miss any of the talks. Today, we are very pleased to welcome Dr. Cristiano Bertolucci, a zoologist mastered by the University of Pisa and a doctorate by the University of Bologna. Currently, he works at the Department of Life Science and Biotechnologies at University of Ferrara. Cristiano does research in animal behavior and chronobiology. His research activity is focused on the study of the temporal organization of physiological and behavioral process, and in particular on the circadian timekeeping system of vertebrates. Dr. Cristiano Bertolucci, thank you very much for your presence here today. After your talk, we will have 30 minutes to take questions from the audience. Everyone can ask questions through the YouTube chat. They can also be in Portuguese if you like, and our team will select and send them to me. This event will be recorded and will be available through YouTube after the transmission. With no more details, I will give to you now. The word to you now. Thank you.
Thank you, Bianca. Thank you for this invitation. Now I share my screen just to starting my presentation. Okay, and take the pointer and it's fine. Okay. Okay. Thank you very much to everybody. Thank you very much for this invitation. It's my pleasure to be connected with you today and spoke about my research activity in the University of Ferrara. Today I decided to speak about my research in chronobiology and in fish, and particularly the study the circadian clock in animals that evolved in extreme environmental conditions. I study mainly fish, not only fish, but mainly fish. And in my department, in the fish house of my department, we breed different species, obviously zebrafish, guppy, also medaka, Japanese rice fish, but also we breed 2 species of cavefish. And now we spoke mainly about the results that we obtained in the last period in this fish, the Somalian cavefish. Why study fish? To study the evolution of the circadian clock. This is an important question for a student. We study fish because fish evolved, colonized, adapted in extreme habitat and in different conditions. Indeed, you find fish in the deep sea, you find fish in the hypogean condition, you find fish in extreme temperate conditions like an Antarctic area. Then they are an excellent model to compare the adaptation to different extreme environmental conditions. I decided to study cavefish. Cavefish is a word that identifies many species of fish that live in different habitats in different parts of the year. In the main study is the Mexican cavefish, Asterix mexicanus, that live in this part of Mexico. There are different morphs of cavefish. It's a blind cavefish, like all cavefish, but these different population that come from different cuevas have different degree of eye degeneration. And the most interesting things of Asterix that there is also the morph with eye. Then we can make a direct comparison with eyeless and fish with normal eye. Other species studies is a species that come from Southeast Asia, Thailandia area, is Balitoridae. Is also have a sleep loss, but maintaining treble circadian clock. But the fish that I mainly study is Phratictes andrewsi, that come from Somalia, is an African cave fish. This is the picture of andrewsi, and you can see this is a Somalian cave fish. They lose the pigmentation and they lose the eyes. It's a cyprinid, like a zebrafish, but he come from this particular area. He live in the phreatic layer in the water table, in the phreatic surface, and this is the area when we collected the animal many years ago in the center of Somalia, in the region called Badbad. This is an Aussie of Badbad, and we collected the fish in the well as we showed here. The animal arrived in this well during the night, probably to eat some things, and then it was very easy to collect this animal. Then this animal living in the phreatic layer of the desert in extreme condition, because evolved in this condition of the darkness, of constant temperature, and very reduced flow of nutrient. And the environment is completely different, for example, from the Asnax mexican environment that evolved in the Karstic condition in these cuevas. When started the cave colonization, we made some genetic analysis studying the DNA of this animal, and we know that the colonization started about 5.3 million of years ago. But the most interesting event that changed the life of this animal was the desertification. The desertification happened 2.5 million of years ago, and after the desertification, a small population of this species was isolated in this condition until now. Then this is clear that the time frame of the cave colonization and complete isolation happened in about 2 million of years. And in 2 million years has been necessary to have this incredible troglomorphic phenotype. Indeed, we know that this is a real anophthalmic animal. We studied the development of the eyes, and we know that some hours after fertilization, starting the degeneration of the eyes, and is very fast. And this is determining complete degeneration of the eyes, reduction of the optic nerve, and reduction of the telencephalic area. The scale are not present. We have a complete depigmentation. You can clearly see from the picture. We have an incredible reduction of metabolism and oxygen consumption, thinking that this fish cannot eat for months without any problem. Also, indeed, they tolerate very long starvation and is very fat fish. Then one of the ideas for the future is also to study the metabolism and the lipogenesis of these fish. But the most interesting things for me is the deep alteration to the circadian clock and the circadian photoreception. Today we spoke about this one. And recently we also investigate the alteration of the DNA repair system, and but is not the topic that we'll tackle during this webinar. Okay, why we decided to study Phratictes andrewsi? Obviously for a zoologist, because it's an interesting model that evolved in this extreme condition, and then it's really interesting to study the evolution of the circadian clock in this animal. But why andrewsi respect to the other cave fish? Mainly because it's great advantage to use andrewsi because it's cyprinic, like a zebrafish. Then all tools that work in zebrafish, molecular, cellular, physiological tools works very well also in Andruzzi. Then it's very easy to study, as it's very easy to use the genome, the transcriptome of zebrafish as reference for our research activity. Okay. This is classical asking diagram regarding the circadian timekeeping system. We know that there are some environmental inputs. There are the entrainment pathway, entrain the circadian clock, and then we have an output. Regarding this output, we have, for example, we can measure, we can measure rhythmic in transcription, in physiology, and in behavior. And the first question that we taken to study this animal was if the cavefish circadian clock can be entrained by Zeitgeber like a periodic food availability. Why we decide to study the periodic food? Because in this extreme condition, in the dark, we're looking for an alternative Zeitgeber to entrain the circadian clock. Then what we do is just to organize an experiment in which at the same time we give the food at the same time of the day in zebrafish and in Andruzzi. And we're looking for the entrainment of the locomotor activity. The results I show you as two actograms. On the left, the Fratictes Andruzzi actogram. On the right, the zebrafish. In both representative example, you find that there is a feeding entrainment because the animal have a period of activity of 24 hours with increase of activity before the meal. Let's say clear food anticipatory activity. These results is also present in zebrafish. And the most interesting things when you put the animal in fasting, we continue to have a 24-hour period, showing that this was a real entrainment and not the masking effect of food administration. We decided to stress this condition, and we repeat the experiment in different group of cavefish and not zebrafish, giving the food every 2 days and every 3 days. And what we found, what happened, that in Fratictes Andruzzi, we found that giving the food, pulse of food every 2 days or every 3 days is sufficient to find a perfect entrainment of the circadian clock. Indeed, when the animal was, after this feeding entrainment, put in fasting condition, we found a clear period of 24 hours, as this mean wave clearly showing, demonstrating that this animal have a strong circadian clock and strong feeding clock. The same thing's not happening in zebrafish. In fact, what's happened during the fasting period, the activity of zebrafish become immediately arrhythmic, showing that the activity that we found every 3 or 2 days is a masking effect of the administration of food. It's not entrainment of the circadian clock. Indeed, we didn't find any clear food anticipatory activity that differently is present in cavefish. In cavefish, we also try to give the pulse of food every 4 days, and we continue to find a good entrainment. This is an extreme condition. And to be honest, I don't think that we can study this, we can repeat this protocol in one different animal. Not, it's probably was typical properties of cavefish. To confirm this one, we decided to repeat this experiment in Astynax mexicanus. Sylvie Reto from the CNRS in Paris, she's one of the major experts of behavior and physiology of Astynax mexicanus. And she gave us some fish, from Pachon Cuevas, to test this protocol. And we found that also in Astenix mexicanus, we found this incredible entrainment. Given a pulse of food every 4 days, we are able to entrain locomotor activity, to find a clear food anticipatory activity, and we are sure that these results is an entrainment of the clock, because when we release the animal in fasting condition, we continue to find a clear 24-hour periodicity. Obviously, we decided to study more than behavioral characterization. Then we decided to investigate at the molecular level, in vivo, in vitro, and ex vivo. Basically, what we do was to entrain Fratistes and Ruzzi at feeding entrainment, give a pulse of food every 24 hours. And after 20 days, we decided to sacrifice the animal at the different time points and collect different organs to test the expression of 2 clock gene, period 1, negative element, a clock 1A, a positive element of the molecular timekeeping mechanism in the brain, in the heart, and the fin for more than 2 days. And what we found in all these organs, we found a circadian rhythmicity of clock, showing that we demonstrate at the molecular level that we have a clear entrainment of the circadian clock. We decided to expand the research to study also in vitro. Why to study also in vitro? Because we want to dissect the mechanism with a series of functional analysis. What we will do with my previous PhD student, Nicola Cavallari, we generate a first cell line from cavefish, from the fin of clayfish. We call this one CF1, and we try to transfect this cell line with the promoter of period 1 plus luciferase to make a reporter assay in vitro, and to recording the expression of this promoter during 24 hours after a pulse of serum, like a shock of serum, to entrain the circadian clock, applying the classical protocol that has been applied in fibroblasts, in mouse fibroblasts. And also we decided to investigate the temperature compensation mechanism. You know that the temperature compensation is one of the mainly characteristic of the circadian clock. We give, then the idea was that the different temperature in the physiological range, the clock continues to have the same period, just to compensate the temperature difference. What we found is 2 interesting results. The first result was that the shock serum was able to activate the clock of this animal. Indeed, we found the rhythmicity of per 1B luciferase report. But the most interesting thing is that we didn't find expression of 24-hour, but find an expression of this luciferase with a period longer. It's where in the infradian range from 38 to 47. And also we found that this clock is not temperature compensated. This is a really interesting result because permit to us to investigate at molecular level this strange circadian clock, because it's clear that P and Ruzzi retain a food entrainable clock, but it's also clear that the cellular level in this clock probably works in a different way respect to what we think, because when we treated with the serum shock, we found a period different to the 24-hour, and we didn't find temperature compensation. What we think to do now is to continue this activity to study the circadian clock at molecular level. We are working to the transcriptome analysis, we think to study the metabolism of these fish. And what we think to study, and this activity is in progress for the next year, because the molecular basis of the feeding entrainment in fish is real complicated, because we know that many different hormones are involved in this activity, like melatonin, orexin, ghrelin, leptin, cortisol. And also there is many different oscillators that are spread in the body in different organs. This is not easy to study. We have the opportunity to investigate the role of glucocorticoid receptor using zebrafish model. In a collaboration with University of Padova, University Polytechnical of Mark, both Italian university, we generate and knock out glucocorticoid receptor zebrafish. And we investigate the circadian clock of this animal. We found that these animals have a perfect light entrainment. Differently, we have an impairment of feeding entrainment. In fact, if you see this actogram on your left, it's a knockout. This is an actogram of a wild type. We found that in the wild type, we have a perfect entrainment to the food, and we have a rhythmic activity in the starvation. Differently, when we work with the same protocol in the knockout zebrafish, we found that the feeding entrainment was markedly dampened or was absent. And in the starvation, the animal was arrhythmic. We confirmed these results also to study the feeding entrainment in larvae. And after 20 days of daily feeding entrainment, recording locomotor activity in the larvae for different hours, for 3 days in constant lighting condition, showing that the wild type have a very good rhythmicity for at least 3 days. Differently, in the mutants, it really dampened. And after 1-2 days, is completely flat. Okay, glucocorticoid receptor could play one of the topic to study to better understand the entrainment to the circadian clock, to the feeding rhythmicity. We hope to develop this part, and I hope to be able to show you new results as soon as possible. Okay, but obviously we did not exclude totally a role of the light. It's clear that this animal evolved in the dark condition, but don't forget that this animal receive some light signal, because when we are able to collect this animal in the wall during the night, but in the wall on the surface, then we think that this animal have some response to light. Then we decide also to study the light as a second Zeitgeber, and just to understand if this animal can be entrained by LD cycle. In pioneer activity, Carl von Frisch, the Nobel Prize in ethology, studied European minon and showing and demonstrate that this animal has encephalic photoreceptor. Because the main question is that this blind cavefish without eyes, which system use this animal to perceive the light? And we know that the fish have encephalic photoreceptor. And von Frisch is showing the first results that minon was able to change the skin coloration, pigmentation. And he hypothesized that the photoreceptor was localized in a specific area of the brain. We know that the deep brain photoreceptor in fish play a role in different topics: skin color change, seasonal breeding, photorefractoriness, molt migration, orientation, but also in phototaxis and movement-related behavior, and the photic entrainment of daily of daily activity. This is true not only in fish, but also in bird, in reptiles. And the most interesting things, one of the most interesting things in the animal that live in the water, that the column of water is like a chromatic filter. Then we know, for example, that in open ocean, very clear water, the wavelength, the short wavelength in the blue arrive in the deeper. Differently in the coastal or clear fresh water, the spectrum is shifted a little bit to the green respect to the blue. Then there is a different light penetration under the water. And this permit us to suppose that is different role of different opsin. Indeed, we know that not only that fish have encephalic and peripheral photoreceptor, they have a photoreceptor in the brain and also in different organs, including the skin. But we also know that in the brain, different kind of opsin are expressed in different areas. In this photograph, in this imaging, it's clear that the different kind of opsin are spread in the brain, are mainly concentrated in the diencephalic area and close to the pineal gland, but it is expressed in different areas. And why have a different opsin expressed in different areas? Good question. Probably one of the answer is that the different opsin are able to perceive in different wavelengths. Then if you have many different opsin expressed in different cells, you are able to respond in different ways at the light spectrum. In fact, it could have some opsin like a parapinopsin PM5 that respond to the violet, to the UV, and the other opsin that respond to the blue or to the green. These are the most important spectrum area for the expression of opsin in the brain of fish. Then we decided to study deeper brain photoreceptor. Then we decided to study photic entrainment of daily activity and phototaxis and movement-related behavior. Well, we study both in larvae and in adult. In larvae, we use Daniovision and Netovision by Noldus, and we exposed cavefish and zebrafish larvae at different wavelength in a classical LD cycle, 12 hours of light and 12 hours of dark. What we found, for example, is here results showing an actogram and a classical graph showing the locomotor activity of zebrafish exposed to LD cycle in different wavelength. And we found that in zebrafish, independently to the color of the light, to the wavelength, full spectrum to white or blue or red or violet, we found a perfect entrainment by day 5 when the animals start to swim. And this is clear that the wavelength is not important for the entrainment of zebrafish. But if we repeat the experiment in cavefish, we found completely different results. Indeed, the zebrafish larvae maintained for many days in LD condition with a different wavelength. We didn't find any kind of entrainment if you use white, red, green, blue, or violet light, showing that this animal, at least at the larva stage, haven't any possibility to showing an entrainment to the circadian clock by light. We repeat this experiment in the adult. As we expected when we exposed a zebrafish, group of zebrafish in different aquarium, in different wavelength, we exposed this animal in LD cycle. We try to shift the LD cycle of 6 hours and we put the animal in constant condition, in constant darkness, independent of the wavelength that we use it for the entrainment, white, red, green, blue, on violet, we found a clear entrainment, we found a clear shift with the transient of the locomotor activity and the new entrainment to the new LD cycle. And when we put the animal in constant darkness, we found a rhythmicity for a period with a period different from 24 hours, demonstrating a clear entrainment of the circadian clock and the presence of circadian rhythmicity. Is interestingly showing that different wavelength apparently induce determining different free running period. But this is another story that we did not investigate. But what's happened in cave fish, in adult cave fish? We found the same results that we found. We find the same results that we found in the larvae. Apparently, yes. This is the entrainment. This is the recording of locomotor activity of a cave fish or Druzzi in LD cycle using white light, and we found a clear arrhythmicity, as we found that we use red, blue, violet, and ultraviolet light. We found continuous arrhythmicity. And these results did not surprise us because this animal evolved in extremely dark condition. Obviously, we was surprised that we will not be able to induce any kind of circadian response in this animal. But these results was also confirmed at molecular, this result was not a unique results in cave fish. In fact, we repeated the same experiment in Asterix mexicanus, and what we found that differently, Asterix mexicanus, the blind cave fish, Mexican blind cave fish, the light was able to entrain locomotor activity. Indeed, using red, blue, and the green light, we found a clear entrainment of the circadian clock, and we also found a rhythmicity in constant darkness. Apparently, the circadian clock of Asterix mexicanus responds to light, is able to be entrained by light. This is a great difference between Asterix and Druzzi. We think to investigate also at the molecular level to be sure about this rhythmicity. Then what we will do is to entrain the fish to LD cycle, then sampled at different time points, different group of fish, cut a small piece of caudal fin and looking for the expression of this peripheral organ, the expression of clock gene. Then we investigate period 1, clock 1A, clock 2. I also investigate some light-inducible gene like cry1A, period 2, and cry5. It's like photolysis. And in all these cases, we found arrhythmicity, clearly showing that the LD cycle was not able to entrain the behavior, but also was not able to entrain the gene expression. But we found one original results, and now we are working on this one. We found the different results if we use the green light. When we exposed the Druzzi to an LD cycle with the green light, we found a rhythmicity, and the animal maintained this rhythmicity in constant darkness. Apparently, the green wavelength is able to entrain the circadian clock. And the preliminary results in terms of clock gene expression in the caudal fin of Druzzi entrained to LD cycle using green light, showing that there is a weak rhythmicity of 2 clock gene, clock 1 and pair 1B. Obviously, these are real preliminary results. Be careful, we must confirm, repeat this experiment, made a sampling of also in constant darkness, but this could be an indication that the green light have a different effect on the circadian clock and induce light entrainment respect to the other wavelength. Okay. Obviously, the circadian clock of Andruzzi lack of normal light input pathway because it's completely different from the other circadian clock that we know, because he apparently respond only on a small spectrum of the light. But this could have some ecological implication that now we don't know, but we must study and investigate this interesting point. We made an experiment to try to understand if the food as Z-Geber were stronger respect to the light. And what we'll do is to compare in a conflict experiment light versus food. Basically, we entrain the fish to try to entrain the fish to LD cycle using blue on green light, and in parallel, we give the food in an opposite phase as we expected. For example, fish was in LD cycle, and we give the food in the mid of the night, and after we shift the food from the mid of the night to the mid of the day. And what's happened, that the animal did not take into account the light entrainment, but take into account the food administration. Then the animal was nocturnal and become diurnal when you give the food. This confirms us that we didn't find any light entrainment, but we found a clear and evident feeding entrainment. But what's happening when we use the green light? Because you remember that we have an effect of the green light. Apparently, the strongest Z-Geber is the food, because when you give the food during the night, the animal was entrained, and when we shift from the night to the light, when you give the food, the animal did not respond very well and did not shift immediately, showing that there is a conflict between the food and the light in the entrainment of the locomotor activity. And these results confirmed us a possible role of the green light in the entrainment, in the setting, the circadian clock. To improve, to deep study this part, we decided to understand which gene could be involved in this light perception mechanism. We tried to characterize the visual opsin repertoire in P. Andruzzi. We performed the transcriptome, the brain transcriptome of Andruzzi, and we found many different opsin. We found complete sequence of exorhodopsin, rhodopsin, conopsin, the type SWS2 is a short wavelength sensitive opsin that perceive in the violet, ultraviolet. We found visual ancestral opsin, OPN3, TMT, OPN5, OPN4, melanopsin, M-type, and Xenopus type. The first results that we obtained, that 3 of these opsin, 1 TMT and 2 melanopsin, M1 and M2, have a point nonsense mutation that results in premature stop codon. Then 3 of these opsin was mutated. And we know that this opsin could play a role in the entrainment of the circadian clock. But apparently other opsin have a normal sequence. What we suppose that, for example, the green opsin like OPN4X, VA, extra-rhodopsin, rhodopsin could play a role in the circadian entrainment to the green light, but we must demonstrate this involvement. And the other opsin could play a role in other behavioral tasks. For example, the photophobic response to different wavelength. Then some of these opsin, like, OPN5, OPN3, perceive a different wavelength, perceive violet, blue, or, for example, exorhodopsin and rhodopsin 1 that perceive in green could play a role in the photophobic response to different wavelengths. We tried to test this one, and then we organized a classic photophobic experiment. Basically, single andruzzi, single cave, Somalian cave fish was maintained in an aquarium. This aquarium had the dark zone and the light zone, and fish were free to move from the dark to the light zone. Then fish was exposed for half hour at a different wavelength. In the first experiment, we test red, green, and blue light. And we looking for the behavior of the animal. We found a negative phototactic response, and fish spend time in the dark zone respect to the light zone. And what we found in these results is that the animal was able to perceive all wavelength, and the animal spend a measure of time in the dark zone. When we reduce progressively the irradiance, then the light intensity, the animal spend more time in the light zone. But what we found at the low irradiance, there is a difference between the green respect to the other wavelength. Apparently, also for the phototactic response, the animal have a measure negative phototactic response using the green light. And this could have an explanation about the quality of the water, that probably there is a filtering of the other wavelength, and the green wavelength is the measured quantity of wavelength that is present in the water, in the wall, the animal moving to go to eat something. We decided to compare the results, to repeat and compare different species. Then we repeated the experiment that we using normal white light, and we compare astinax with the cavefish and with the zebrafish. And all these three species showing a clear photophobic response to the white light. The most interesting thing is that if we use different UV light at different wavelength, we found that we have a clear photophobic response for Somalian cavefish and for zebrafish. We haven't this response in Astinax mexicanus. We don't know exactly why. We must investigate these things, but these results are clear. Then basically these results showing that some opsin expressed in the brain of cavefish could play a role for the circadian treatment. Other opsin could play a role in other fundamental behavioral response, like the phototactic, to escape from the light and to escape from predator. Then it's clear that the functional connection and the role of the deep brain photoreceptor is crucial, is important in endocrine, circadian, and also metabolic process. But this is remain unclear. We must investigate to discovery of the role of this deep brain photoreception. And the cavefish that evolved in the extreme photic environment are a powerful model to study this no visual photoreceptor system that, don't forget, that probably is the first photoreceptor system apparent in animal. And it's also important to study the evolution of many pathways, many processes in response to light. One thing that we could think to study as other environment with low and no rhythmicity. One possibility could be to study deep sea habitats. Why deep sea habitats? Because the animal that evolved in deep sea habitat probably have a similar situation of the cave fish. Then could be interestingly to study clock gene expression and no visual opsin, and theoretically some circadian and daily rhythmicity in peripheral tissue, but also at behavioral level in species that live in this aphotic zone. This is one of the ideas that we have to study in the future, continues to study cave fish, but maybe in the future we'll be able also to study deep sea habitat. Well, I must thank my research group in Ferrara, then Tyron Coxicato, Stefano Grignoli, and Elena Fregato and Elia Gatto that work with me. They are involved in different topics, but they study circadian clock at a different level, more behavioral, more cellular and molecular, and also in other wild conditions in the animal. And also must thanks my student, master student, PhD student that works in my research group, that we collaborate all together to perform these results. I show you only one of the main research activity that we developed. And finally, I must thank all my international collaboration, like Nick Folks at the Karlsruhe Institute of Technology and the Center for Organism Study in Heidelberg. Nick and I developed all research activity in Abruzzi, and the CNRS in Paris that help us with study in Astyanax, and the University of Murcia, Francisco Javier Sanchez and José López Olmeda, and we collaborated with feeding and entrainment in the circadian clock, and also the Stazione Zoologica Anton Dorn Napoli that give me a fellow and the possibility to study everything. Thank you very much for everybody, and I hope to see you soon in Ferrara, one very nice town in the center of Italy. It's classical middle-aged town. Let's see, very nice to live here. This is our campus, and you are welcome in Ferrara. Thank you very much to everybody.
Thank you for the amazing talk. Now we have some questions from the audience. The first one, we showed that the free running period is far different from 24 hours at a range of temperatures. Have you tried to entrain with 24 hours food cycles at these different temperatures?
Not yet. Is one of the experiments that we perform next year. We are working to realize an aquarium in which we able to the fish to choose different temperature, like a behavioral selection of environmental temperature. And we did not put any conflict between food cycle and the temperature. This is one of the activities that probably we performed next year.
Interesting. The second one, does the water temperature changes cyclically in the natural environment?
This is most interesting things. Probably the temperature did not change. Probably is stable. They are around 20, 25, 27 Celsius degrees, the water in the phreatic layer, and we think that the temperature is constant, don't change. Then I think in environment, we have a constant darkness and a constant temperature. This is an extreme environmental condition. And if temperature is one of the Zeitgeber that the evolution used to set the clock, this could be an extreme condition to study this situation.
Oh, nice. 3, in your study, in your studies, it was shown that cavefish clocks could work if given a relevant signal, such as food. Beside the photoperiod, it's possible that this stimulus synchronizes also seasonal changes?
This is really good question. In Abruzzi, I don't think that it's the best model because we are at the equator. Then the LD cycle is constant and also evolving in the dark. If I can, I study now, we have a research project in collaboration with University of Heidelberg, Joachim Wittbrot and Nick Folks. We study Medaka. Medaka is the Japanese rice fish. This is a real seasonal animal. And what we found, we tried to put in conflict food and light with different photoperiod. The results are really interesting and very, very preliminary. I have the impression that during the winter season, in which there is a reduction of the activity, the effect of food as Z-GABR is reduced with respect to the summer condition, which I might have a high metabolism. But this is only speculation. We must increase the test, not a behavioral level, but a level of metabolism, metabolic analysis.
Oh, interesting. Let's see. Have a temperature. Oh, sorry.
No, yes, please. The last question, the last 4 questions for this.
Oh, I lost here. Oh, sorry. Have temperature cycles being tested for Z-GABR? Oh, yes, I always have problems with those words. For these cover fish?
Not yet. As I replied you before, it's a program for the next year. We made some preliminary tests in the cell cycle and apparently works as in training agent, but it's a preliminary results in cell cycle. It's not the whole animal at the behavioral level.
Oh, interesting. Cristiano, thank you for your talk. Very interesting data. Are there other no photic cycles that you would expect to enter in the cave fish, like food available did? For example, social entertainment?
Allora, social entrainment, I don't know. Obviously, temperature, we spoke before. Social entrainment, I don't know, because when we're looking for the behavior of the animal in the aquarium, we have a big aquarium of 400 liter, 300 liter, in which we have, I don't know, 20 fish, 30 fish. We didn't find any shoaling. Each animal move swimming independently. I don't think that there is some social response, but a specific test on the sociality we never performed. This could be a good idea, but the social as entrainment, I don't think so.
Interesting. I've seen some discussions about the synergic and independent action of oscillators like the light-dark cycle and food. Some authors suggest that food can be a clue that overcomes light-dark, while other authors believe that no clue overlaps the night cycle. What do you think about it?
Oh, this is a really good question. This is like, it's really complicated. I think that in some condition, the food is more important than light, as Z-GABR is the strongest probably. I think that the mechanism is different. The nuclei, the encephalic part, and the gene, the hormone, the molecules involved are different. I have the expression that we have a light entrainable clock and the food entrainable clock. And these mechanisms are independent, are not together. Probably they evolved together using different pathway to arrive at the same results that entrainment of the circadian clock. But I have the impression, in my experience, that two independent pathways. I don't know if I reply to the question of Giovanna.
Oh, interesting. I guess we have no more questions. All right, let's see.
Okay.
Do you think we can infer any aspects of the evolution of biological clocks in vertebrates by studying fish and how they respond to different wavelengths at the light spectrum?
Okay, I think yes, because obviously they evolved in particular environment like in the water, then it's water is a chromatic filter. Then we have different photic environment respect the subterranean area. But it's the first vertebrates, more or less the first vertebrates. Then it is crucial to study the evolution of the circadian clock. Furthermore, don't forget that, for example, when we studied zebrafish, 70% of a genome of the gene the homologous gene are also present in the mammals. Then we can think that this gene could play the same role. Obviously, the evolution, something happened. They have different adaptation. Looking for the expression of encephalic photoreceptor, they are crucial in non-mammalian vertebrates. Probably they are not really crucial in mammalian vertebrates, but now we know that probably they also play a role also in mammalian vertebrates in some specific topic. But I think that is crucial in terms of comparative analysis and to evolution to study fish to arrive to mammals.
Oh, great. Great talk, Cristiano. Thank you. You showed the patterns of expressions of opsins in the brain. Is that pattern conserved along fish development, larvae to larvae from adult, and are between different fish species? Okay. Between the different species, more or less, the pattern is the same. Obviously, we have a high expression of different opsin one respect to the other, different location of the opsin. During development is more complicated. It's more complicated, and I think that systematic data showing different location of different opsin in the brain of larvae and juvenile and adult during development, this part of the study. But this could be very, very interesting things, like study the development of, for example, phototactic response in fish. We try to study this thing, but it's a really complicated larvae. It's not easy to do, and also as all things that are continuously in progress each day during development, zebrafish changes until, I don't know, until become adult. Then it's not easy to do, but I think that this could be one interesting things to study. Oh, nice. I can imagine. Now, I guess we have no more question. Let's see here. No. Thank you very much for this. I guess we have no more question. Okay. Thank you very much for this amazing talk, Dr. Bertolucci. And also thank you for the audience for all their attention and questions. I hope you two have enjoyed this talk, and see you next talk. Okay, thank you very much for this opportunity, and good luck for everything. And I hope to see you soon. Bye-bye. Thank you. Thank you to you.